to improve research skills among our students. After this workshop, if you feel like you would like to practice some more, check out our call for a paper review competition on our social media platforms. In terms of communication during this workshop, please feel free to directly ask questions in the chat, or if you would prefer to speak, please type an I for a new question or a new point, and two I's for a direct response to what is being said, and I will give you the word as soon as possible. Joining us today is Ms. Mariana Barbosa, a postdoctoral researcher who has been working on screening the biological range of potentialities of marine natural products at the Laboratory of Pharmacognosy of the Faculty of Pharmacy of the University of Porto, and Mr. João Bernardo, currently concluding his PhD in Pharmaceutical Sciences with a specialization in pharmacognosy, who is mainly working on the characterization of the phenological profile of extracts obtained from medicinal, also at the Faculty of Pharmacy. I would like to thank you both for taking the time to host this workshop. And when you're ready, the floor is yours.
We are going to start sharing the screen, okay, Mafalda?
Mafalda, you need to make us able to share the screen.
Please try it now. I think you can now.
Yeah, okay.
Okay.
Yeah.
Oh, sorry. Okay.
Okay. So, hello everyone, and welcome to the workshop How to Draft an Abstract Interpretation of Results. So, as Mafalda already said, I'm João Bernardo, and with me is Mariana Barbosa. And first, we want to thank for this opportunity to share some experience with you, and we are very glad to collaborate, okay? We are both young researchers, and we are developing our work at the Lab of Pharmacognosy. And today, we are here to share with you some of our expertise, some tips and advices, and how to draft a research paper, and especially focusing on some key points to improve your research skills. So first, we would like to introduce you to our lab, the Lab of Pharmacognosy. We are located at the Faculty of Pharmacy of the University of Porto in Portugal. We have a very beautiful country, and if you have not visited yet, we hope you have the opportunity when this pandemic is over. Here you have let me point. You have an aerial view of our facilities, and you can see that we have a great landscape of the Douro River. Also, one of our icons is the stained glass from 1934 that celebrates the pharmaceutical tradition in Portugal. And we are very proud of our faculty. In fact, this year, we are celebrating 100 years of existence. And also you have a picture, a photo of some members of our research team. We have some master degree students, some PhD students, of course, researchers and professors. And in the center, you see Professor Paula Andrade, and she is the scientific coordinator of our group. Okay. Research in pharmaceutical sciences is quite challenging, and this is because it often comprises several major areas of knowledge that goes from chemistry, physics, biology, microbiology, and of course their application towards the discovery of new drugs and development of new pharmaceutical formulations. So it takes some time to acquire all the necessary skills and know-how to perform and interpret the results.
Our group at Requinte, which is the network of chemistry and technology, has been devoted to natural products, the chemistry and bioactivity. So this group comprises a cross-section of several scientific domains that are really categorized in some sections, like in this pipeline, like we present you here, this clinical pipeline, this research and development pipeline. So the group activity includes usually the selection of natural matrices. In fact, over the last 20 years, our group has been investigating more than 300 natural matrices, including plants, algae, microorganisms, insects, marine invertebrates, a series of natural sources. And in fact, we have been focused on the extraction and isolation of bioactive compounds from natural origin, with the evaluation of their therapeutic potential within, of course, the scope of this preclinical research, okay? And this is which we'll be showing you today some examples of our work in this area. Of course, we are also able to identify some of the major compounds that can be responsible for the observed effects.
Okay, so despite the particularities of each field, the scientific method is common to all research projects, and it is a well-defined process that guides the researcher through work planification in order to solve a problem. And it starts with the observation of a problem. Okay, so you formulate a question and it has to be solved. And so we formulate hypotheses and test them in order to obtain our answers. And when we analyze the obtained results, we can define whether the answer is correct or incorrect. And if the hypothesis is incorrect, it's necessary to go back and elaborate a new one and test it, of course. But if it is correct, then we can draw the conclusions. And when we get the satisfactory level of conclusions that will support our answers that were posed initially, we can move on to reporting the results. One very didactic way to demonstrate to you how the scientific method works is this example that you can find online, which is the failure to toast. You start with the observation, the toaster won't toast, number one, and then you ask the question, why won't my toaster toast? So if you formulate one hypothesis, for instance, maybe the outlet is broken, okay, number three. And you make predictions, well, maybe I plugged the toaster into a different outlet, then it will toast the bread. And you test it, of course, in order to verify if it's true. And so you plug the toaster into a different outlet. And then you analyze the results. If my bread still won't toast, then it's time to formulate a new hypothesis. It was not supported, of course. But if my bread toasts, my hypothesis is supported. The outlet was broken. But then you have another question. What is wrong, or what was wrong with the first outlet? As you can see, this is always what we do in science. In science, answers always open new questions, okay? So after doing this fun cycle so many times, exhausting sometimes, we will finally get our answers to support our hypothesis and answer our problem. And one of the most important parts in science is communication. And scientific communication can be done in very different ways. For instance, PhD thesis, or an oral communication in a congress, or a poster communication, or simply a report that you have to present to your professors during classes. Well, regardless of the type of report, there are always some shared parts, or some common parts. And one of the most important is the abstract.
Yeah, this is really the main topic of our subject today. So abstracts, in general lines, of course, they are usually independent and short texts, and usually they do not should exceed the 10% of the overall manuscript. Uh, and the main purpose, the main objective of an abstract is really to capture the essence of a paper and also to raise the reader's attention to a particular subject. So a good abstract, a perfect abstract, should also raise some curiosity level, some excitement, and therefore it should contain some powerful sentences, some powerful statements that really highlights the scientific novelty of the research you developed, okay? So overall, an abstract should include the main findings of your research work and also some key questions for further discussion, okay? So writing a good abstract involves always knowing the rules, the rules of the paper you've chosen for your publication. So you have to follow, you have to check the journal's guidelines, and in fact, the guidelines of a journal vary a lot from publication to publication. Of course, you have to use the right language and produce or attempt to produce a flawless text, of course. On the other hand, you really have to have good summarizing skills. Okay, so you can provide a general picture, an overview, a general overview of your work, but also highlighting your main arguments and topics. Okay, so we show you here some examples of traditional, I think, abstracts. And as I already told you, even though it is important to elaborate an appealing abstract, of course, it is also important to keep it concise and factual. And of course, references or abbreviations should be avoided in this particular section of a publication. So as I was saying, this is an example of a common abstract with word count, okay, with the limitation of the number of words that you can write. Normally is 250, but some journals, in some journals is usually less.
100.
Yeah, 100, yeah. So you have to practice your summarizing skills a lot. Sometimes, okay, but even though we try to respect the overall organization of an article, in which you have your introduction, where you point the research problem, your hypothesis. We have a materials and section, okay, with the methodologies that you use for your work. With the main results, the main findings, and of course you have to highlight some conclusions that you reached with your work, of course. And be careful, because sometimes this is the only part that some editors from the journals read. So you have really to highlight your abstract. And sometimes you will also be asked to provide a graphical abstract, like this one here. So a graphical abstract is also called sometimes a table of contents graphic, okay, which is also supposed to summarize your work, but through a figure or a scheme, okay. Unlike the general figures within a manuscript that are usually black and white, the use of color is always encouraged in a graphical abstract. It should also be simple but informative, and most importantly, it should not contain any graphic information or results that you have in your paper. And this is why we ask a lot why. And the practical answer is the graphical abstract is usually free of charge, okay? When you access to a graphical abstract, you don't have to pay to access to that. But when you have to look into a graphic or a chromatogram in a paper, you have to pay for that access, okay? That's why you should not add your results to the graphical abstract. In here we have another example, and this abstract was written by João. João tried to publish, and he was really good accomplished in this Journal of Ethnopharmacology. And as you can see, this journal has a specific type of abstract, a structured abstract, in which you have to present in separate topics your aim of the study, your materials and methods, the results, and the conclusions. So you don't have really a word limitation, but you have to present the abstract in this structured way. But for João to publish in this journal, he needed to add these lines here, in which you have to present the ethnopharmacological relevance of the plants he selected. I don't know if you are aware, but ethnopharmacology is like the study of the empirical use of a medicinal substance that is used, I don't know, in tribes or indigenous populations, okay? So we are, in our group, we try to provide some scientific validation for the use of some plant species in the other development countries normally. Okay, so.
Okay, so now that we already understand the importance of an abstract, okay, and how it will make sometimes will make it publishable or not. But of course, to write an abstract, we need to have good scientific work behind it, okay? So what we are going to share with you now are some suggestions and strategies about how to plan your work, obtain your results, and then interpret them, and of course, write a good abstract. Since you cannot be here in lab with us, we would like it a lot, but it is what it is. We will analyze some of our already published work, okay, with you to give these tips. Okay, so of course, our research work starts with planification. Okay, and for that, we have to define the field of study. And for a student, it may go from a topic that you have been assigned to, or just by choosing the laboratory where you will develop your work. Logically, you have to define the problem or the question that we will explore, and you can imagine that a large part of research goes through literature review. Well, why? Because it will help us to know what was already been investigated, what are some possible new perspectives or the new questions that can be raised. And for that, we can easily access some databases such as Scopus or Google Scholar. It's free database. And then something that it is extremely important. It is necessary to outline the research plan. Here we present you a flowchart. It's quite an elaborated one, but it's not necessary to do something like this, okay? But the advantage of the outlining is that you are able to easily put your ideas in order, organize the steps that you are going to take, and you can also identify where some problems may arise and how to overcome them. And also, if you want, you can put in each section the materials that you are going to use or that you are going to buy, okay? And also important, it's a very appealing way for you to present your ideas, okay? When you are discussing with your research team or your professors, and it's a good way to boost the discussion, okay?
Our group and both of us, we have been particularly focused on the study of phenolic compounds from different natural sources. I personally work with marine sources, with seaweeds, and João works with medicinal plants, okay? And we also aim to screen their range of bioactivity. But for these, there are some key points regarding especially sample selection to extract and to obtain our final target compounds. In the particular case of phenolics, of course, we have to ensure the proper storage conditions and handling conditions to avoid compound degradation, of course. Also, proper extraction with solvents or a mixture of solvents will allow us to obtain a large amount of these compounds, which is a big problem in the area of natural products. And that's difficult than the isolation of some compounds, which is an expensive and time-consuming process, and sometimes extracts or purified fractions or rich extracts arise as valuable alternatives. In that sense, our work has been mainly devoted to extracts or purified fractions. And in here, of course, I'm not going to explore this point by point, of course, but you have an example of an extraction protocol, which is accompanied by a cleanup steps that will allow to obtain purified extracts in the particular class of phenolics that exists only in seaweeds, in my case, which are phototanins.
Here, as Mariana said, I work with the medicinal species. There's also another example of how to extract phenolic compounds from a natural source. In this case, from elderberry. I don't know if you know elderberry. They are quite beautiful and tasteful. The extractant here is water. It's not a solvent, organic solvent, like Mariana performs its extraction. And one of the disadvantages of working with water is the elimination in the final steps. This step is easier when you extract compounds with organic solvents because they easily evaporate, okay? However, one way to remove water from our final product is by lyophilize the extract. And it also has some advantages, why I like so much to use water extracts. Well, it has high yields of extraction. It also allows to preserve the phenolic composition, and the extract is then easily redissolved in aqueous buffers. And these buffers are the ones that are present in most biological assays that we use to test the biological activity of our compounds or extracts. Furthermore, it also allows the cleanup to obtain purified or semi-purified extracts by solid phase extraction. And if you want to do it, it's something that it's easy. For example, if you use a C18 column, and through loading and diverse activation steps, you can clean up the extract and separate the compounds in different parts. Okay? And in this case that we show here, with this protocol, we easily manage to separate some unwanted phenolic compounds from anthocyanins. And anthocyanins are the phenolics that are present in this extract and give that color characteristic, that pink reddish color that you see. I'm going to show you also another example. And in this case, it's a protocol for extraction of compounds also from a natural source. But here, to obtain phospholipid rich fractions from egg yolk. Okay, so we start with the egg yolks, and what we want to have finally are phospholipid rich fractions. And this is just a simple way to exemplify how a flowchart not only facilitates the interpretation of the results, but is also a good way for organizing your mind. Because as you can notice, that is an extensive protocol in which the different fractions are obtained and then submitted to different steps. So it increases the probability for errors to occur, of course. And there is no doubt that a schematic representation of this extraction protocol facilitates your work and also eases the interpretation and replication by other researchers that we will follow your steps. And now that we have our compound separated or isolated, one of the next steps that we need to do is the identification and quantification of the compounds. And of course, there are several techniques that can be used. In our lab, we often use chromatographic techniques. And how do we move from the technique to the interpretation and representation, discussion of results? Well, the first thing that you should do, and this is you should always do, is literature review again, okay? It seems boring sometimes, but time consuming, but in the long term, you will see that as huge advantages. And why? Because you can check if this is the first time that these compounds are identified, or what were the techniques that were used and the correct solvents, if they were already extracted from the same source or from another materials, and if you can validate your extraction and the future identification of your compounds with available commercial standards. And if you're lucky, not so many times, it can help you a lot, okay? If you are working with phenolic compounds, you will find that are generally used hyphenated techniques such as HPLC-DAD, that are used to do the identification and quantification. And you can immediately get some important information. For example, the type of column that is used, possibly a reverse phase column. Also, the composition of the mobile phase, which means the type of organic solvents that you are going to use, if they use water or not, acidified water generally, and if they use a solvent mixture, and if the chromatography is carried out on a gradient or an isocratic elution. And after injecting the samples in the chromatographic system, we will obtain the relationship between the elapsed time and the signal intensity that is registered in the detector, recorded by the detector. Typically, in our lab, you use a UV visible or mass detector. Okay? And for that reason,
you have a question in the chat.
Okay.
Do you want me to read it? What if you are the first to extract a substance? How do you validate the method?
I'm sorry.
Can you repeat it, please?
What if you are the first to extract a substance? How do you validate the method?
We usually validate the methods. We can do by two normal ways, general ways. Of course, in literature review, sometimes there are already standard extraction protocols that are validated, okay? So when you do your own extractive protocol, you can compare it with it, okay? Compare the extraction yields, for instance. Also, you can validate your extraction by adding an external compound, okay, by adding a standard. And that is, of course, not present in your sample, but as a similar chromatographic or extractive behavior. And then you can see that if that compound that you already know and it's validated is extracted with your own compounds, you can validate that the protocol is efficient.
Yes, the validation, you have to do also the recovery, the intradays. You have a lot of work when dealing with the validation of an HPLC method or a GC method, okay? I don't know, Alan, if we asked your question, but that's it. You have to follow sometimes you have to do intradays with different injections in different days, okay, or in the same day. You have to measure the recovery, like João said, with an external reference standard, yeah. That should not be on your sample, but it should be similar to the compounds.
And compare with the previous standardized protocols. Of course, if there is something new that no one has never performed, it is the first time you have to explain your process. But it's generally someone else has already done some type of work that can fundament your protocol, of course.
And of course, we cannot forget the NMR.
Yes.
And the compound elucidation, of course, that is the most valuable thing when we are trying to characterize a sample. But natural matrices are so complex, so it's really difficult. Alan, did we answer to your question? I think so, Mafalda. Can we move on?
I think so too.
Okay, thank you.
Something we will return.
Yeah.
Menção atrás.
Sorry.
Okay, so as you can imagine, we have here the table, but we will generate an immense volume of data. And the most typical form of representation of all this data is the construction of a chromatogram in which each peak may correspond or not to a single compound. Okay. At the same time that we record the retention time for each peak, that once again may or may not correspond to a specific compound, there is other information that we can also collect. And for instance, we can start a table in which we also record the characteristics of the UV visible spectra and also the observed mass fragments. Okay. In this way, we can relate all the data obtained for a possible compound and then try to identify it. We have here two examples of two compounds that were identified in this extract, compound 1 and compound 11. And if we get the correspondence between the characteristics of these compounds with those of commercially available standards that are also analyzed under the same experimental conditions, we can finally identify or potentially identify our compounds. Sometimes we have to say that standards are not available because they are not, or just because they are extremely expensive. However, by comparison with previous works, we can all that with the chromatographic conditions are the same and were reported, of course, we can also try to tentatively identify these compounds. Let me show you another example of a phenolic profiling of an extract, and this time also with the elderberry extract. And it is just to demonstrate the importance of the representation of a chromatogram. As I told you, the spectra are registered by a detector, and in this case is a DAD detector, which means that each spectra is recorded at multiple wavelengths. Okay, you have here 280, 320, 350, and 525. And what is the advantage? If you notice this chromatogram, some compounds are visible at some specific wavelengths and not in others, okay? Practically undetectable in others. And so, if you were simply analyzing the results in a single wavelength, we would misinterpret the results and we would not be able to identify some compounds or just simply misidentify them. And logically, each spectra is related to the structure of its respective compound, of course. In terms of quantification, once the compounds have already been identified, you can use external standards to quantify them by interpolation with external curves, of course. Or if you don't have the standard, again, we can use other compounds that has a similar structure and chromatographic behavior, as we explained in the validation of the method. And of course, these are necessary demands because, as you see in the previous slides, you need to have the similar behavior, absorption, mass, and chromatographic behavior, of course. Now that we already have the results, how can we interpret the results? What is important to highlight in the abstract? For example, how many compounds were identified? Was it the first time that someone did this? Are there some chemotaxonomic markers that validate our initial raw material? Or for instance, the total quantified mass and extraction yields. These are some examples, but of course, this is some important information that you can highlight in your work.
Yes, and João is very lucky to work with medicinal plants, okay? Because I work with a specific class of phenolic compounds, which are fluorotanins, and fluorotanins are exclusively produced in brown seaweeds, okay? So Portugal, as you know, we have a large coast, and the aim of my work is to try to add some value on our national natural resources. of course, the fluorotanins, I have established the composition in terms of fluorotanins of several seaweed species from our Portuguese coast. And I have used some of the techniques that João highlighted before. But when you have a class like fluorotanins that is not so much studied, like phenolic compounds from plants, you have the problem that João was speaking about, the lack of commercially available standards, okay? So most of the works have been characterizing fluorotanins in a natural matrix by quantitative assays, by colorimetric assays, okay? But the problem is we can be overestimating or underestimating the amount of the compounds. And although these assays are quite simple and cost effective, okay, they provide no information on the real composition of your sample. So we use these hyphenated techniques, of course, these advanced mass spectrometry-based methods. And in fact, as I was saying, and as there is no available standard, commercially available standard for fluorotanins, we have to go to literature and find, and we also only can characterize the fluorotanins by their characteristic mass fragmentation, okay? And of course, today is not the day I'm going to evaluate your chemistry, your knowledge on chemistry, forget it. But I'm going to show you just an example on how we perform this kind of identification, okay? So in here you have 373 in terms of mass, and I'm going to tell you some information, some basic information, like fluorotanins are polymers of this molecule, the fluoroglucinol, okay? And this molecule here weighs 126 Dalton. So in here, by this information, we can assure that our molecule has 3 fluoroglucinol units, okay? So we consider it a trimer. Yeah, there is no identification, okay? It's a trimer, 3 units of fluoroglucinol. So they can be linked to each other by carbon-carbon bonds or carbon-oxygen carbon bonds, okay? So I'm not going to ask you because I think this is really basic, but of course it is more easy to break this bound here than this bound here, okay? So we have some characteristic mass fragments that I'm going to show you now. We have this one, which is the exact value that I was telling you of the fluoroglucinol unit. And we also have this one, which can be the fluoroglucinol with an oxygen atom, okay? And as I was saying, this is more easily to break than this one here. So we have this fragment in the MES3 that corresponds to this group, this Foucault group. Okay, so as I was saying, this is a Foucault group, okay? We have an intermolecular break, yeah, fragments. So this is a Foucault group. So our compound is named a Foucault fluorotol, okay? So I know it was 2 minutes to explain this, yeah, but
it seems easy, but it takes a lot of work,
yes,
to identify the compounds. It's, yes. And Mariana, it's very good at this point.
Thank you very much. So, as João was saying, our characterization at this point is merely tentatively, okay? So we have to do isolation, of course, and NMR elucidation to reach the full characterization of a sample. But look at this one. This has 22 compounds, and the peak resolution is not that amazing, okay? So we have another question, Mafalda. Aren't there?
Yes, so
Yes.
Yes, the thing is, it can be a good thing or a bad thing. It can be hell and heaven at the same time. First, because you will find that a lot of the fragments are present in numerous types of compounds. Okay, mass fragments and spectra, UV spectra, are similar between a lot of compounds. So sometimes if you have a library already in your informatics system, it's very easy for you to go and click and check, oh, the maximum absorption is 280. Yes, it is the same for almost every compound that is phenolic, okay? So, yeah. And then you go, okay, but we can add some mass information. Yeah, but the fragmentation patterns are very common, and that is a good thing because, as Mariana said, when you look into this structure, we can see that it's easier to break the structure in that oxygen position than in a carbon position. So it allows us to make the puzzle, okay, together. So yes, there are some softwares, of course, but in our lab we are always used to validate the compounds with the standards, external standards that are injected in the system and the same conditions. Because you not only need to have the same retention time, you need the same UV spectra, you need the same mass spectra, and if you could really isolate the compounds and perform an NMRA or other techniques, that would be perfect. So yes, there are some systems, but be very careful when you use it, okay?
Yes, we use some libraries, okay?
But we also have some
yes.
previous knowledge that make us orientated in what to search, okay? Because you can sometimes have a very similar UV spectra, but you have a retention time different in 30 seconds, and you know that is not the same compound, okay? Just for that. It's like the spectra are so similar, but then you have different retention times. And you can also, of course, say that under the same conditions, those are not the same compounds.
You can have a difference in 2 or 3 nanometers, nothing more, okay?
It's not, so it's hell and heaven for you. Of course, it's a good tool. And when you already have literature review and you see that some compound was already identified, okay, so if it was already identified, if I have the same characteristics in this peak, maybe that is the compound. But have to inject the standard if you have it or you can buy it to validate.
And this information is really important for you when you write an abstract, okay?
Yes.
Because you can say, we have identified 22 compounds, okay? That information is really
good.
It's good, okay? But you can also say, we have attempt at identifying, or we identify tentatively 22 compounds with the spectrum of phenolics or something like that, okay? So it also depends on the techniques that you have available, of course. When you have just an HPLC DAD, VAD, of course, you cannot reach to that conclusion. That's why we always say when writing an abstract, okay, powerful statements, yeah, that's really nice, but
make sure that are correct.
yes. It has to be factual.
Exact. Okay, the information that you provide. It's not bad to say that you tentatively identified, but when someone is going to take your work and start to understand it, to explore it, he will say, okay, he tentatively did it. The information is correct.
But he was not able to achieve it.
So it's the next step for someone else.
Okay, so moving on, Mafalda, okay.
Okay, so we talked about the identification and quantification of phenolic compounds and some information that you can possibly highlight in the abstract. And if you are working exclusively in the chemistry, we would probably already have good material to present or discuss. However, we also focus our work on assessing the biological potential of the compounds or extracts that we obtain. And for that, we often use in vitro assays because in general, they are very simple, they are cheap and fast, and allow us to evaluate the interactions of our samples with some biological systems or models of biological systems. As you already know, phenolic compounds are well known for their antioxidant potential. And one in vitro assay that we can use to measure the anti-radical activity of our samples is the scavenging of the Dpph radical. So it is a stable free radical, and when a compound donates a nitrogen atom, it gives rise to the reduced form of it with the loss of violet color. So the first thing to check is, of course, when you're starting the work, for possible limitations. And you can see here that Dpph is solved in methanol. So we need to ensure that our samples are also able to be solved in methanol. Otherwise, we will have suspensions and not solutions, okay? And we always work with solutions. And if we have suspensions, the samples will precipitate during the assay and our results are not concordant and they are not correct, okay? In addition, of course, as for extraction protocols, it is also important to add a positive control to validate your work. So here we have the schematic representation of the Dpph scavenging, and in this case we selected ascorbic acid as the positive control because it is not present in our samples. Don't forget it. Okay, and the first thing you need to do is to make dilutions. And remember that we want solutions and not suspensions. So if you need it, you have to work in a lower range of concentrations to prevent the effect of the precipitation in the wells. So we start the reaction, and after a defined period, we proceed to read the absorbance, and we register it in the table that you see below. And we can verify in this case for the positive control, that is the ascorbic action, that we have some different coloration, okay, in the wells. And in the control line, the column, sorry, where the reaction occurs in all its extension, we have higher absorbance values than in the other wells where some concentrations of ascorbic acid are present, okay, where we added also ascorbic acid. So when we plot the results, we will find this graphic, okay, that relates the concentration of ascorbic acid with its effect. And what we can see is that we observe a concentration-dependent effect. It's the same to say that in this case, higher concentrations of ascorbic acid are more efficient to scavenge the Dpph radical than the lower concentrations. And one thing that you can also calculate is the concentration where ascorbic acid displays 50% of the effect. That's what we frequently call IC50. And this is a good indicator to compare with other samples and of course to highlight in your report, in your abstract. And then under the same conditions, okay, the exact same conditions, you can test because we already validated this methodology, you can test other samples that you have, that you see in the other graph, okay? And you can see that these two extracts, of course, they have IC50 that are different. And don't forget that a lower IC50, it means that the sample is more potent, okay, than the higher one. But what is the relevance of the Dpph scavenging? You have to be very minutious and to question all your work, because the Dpph does not have a physiological relevance. It's not present in your cell physiology, in your body. So are there any other assays, complementary information that then you can support your results?
Yes,
Yes.
there are. But yeah, like João was saying, Dpph is an important tool, but is like a preliminary tool when dealing with phenolics. But we have other free radicals that are biologically more relevant, like this one here, nitric oxide. Nitric oxide is usually involved not only in oxidative stress, but also in the inflammatory response. So when we aim to scavenge this radical, this biological radical, we can modulate either oxidative stress events or inflammatory events, or even both, which are in fact usually connected. So in here we have like for Dpph, we have the experimental set of this assay, of course. But what I really want you to focus is that from this amount of work, of course, when you are reporting your results, you should focus on the IC50 or IC25, okay? And this is really important. Like for me, I was working with 4 samples from different seaweed species, okay? So this is really important for me because from the IC50 I could establish which sample was the most promising at inhibiting or at scavenging, okay, these radical species. And so I can select the most promising extract or the most promising species for another assays, okay? And that is exactly our group deals a lot with this kind of in vitro assays, the enzymatic ones, okay? So we are trying to increase the biological complexity of our research to validate our results and to because this is in the framework of preclinical research, but we can move on to in vivo assays and finally to humans. So our work as our group has a vast know-how and expertise in these enzymatic assays, and we can determine whether an extract or a compound is able to modulate the activity of several key enzymes involved in several pathological events, namely in diabetes, in inflammation, or neurodegeneration. Today, I'm showing you an example of an enzyme that is usually related with the skin conditions. Okay, tyrosinase is an enzyme that is involved in the synthesis of melanin, but these days some research are pointing out the involvement of tyrosinase in the synthesis of neuromelanin, which is linked to some dysfunctions or degeneration characteristic of Parkinson's disease. Okay, so this is a really important assay that both I and João started and validated yes, in our lab. So and it is a really simple assay, but the enzyme is really expensive. And we are talking about an enzyme isolated from mushroom, okay? Because the human one is 10 times expensive. So in here you have the representation of the assay. As you can see, the color is not that native as with the Dpph one. So because we have a spectrophotometric read at different wavelengths, okay? And once again, we can see a concentration-dependent response, and we also can compare the potency of our extracts. Before I'm going to the next slide, Mafalda, I think we have another question.
Yes, from Alan. I don't know if he wants to ask it by voice because...
Sorry, but let me...
because he lifted his hand, but he asked, how is it linked? Does it cause it or prevent it? I think about the neuromelanin and Parkinson.
Yeah, okay.
Okay.
Yeah, yes.
Just to say that this is again a double edge, because initially this mechanism is one that is used to detoxify products. Okay, the production of this neuromelanin, it's a normal way to detoxify toxic products in our brain cells. However, when it's highly extensive, it occurs too much, it will lead to, may lead to pathological conditions.
Yes, the accumulation of neuromelanin in the substantia nigra, okay, that is one of the, has been linked to Parkinson-like symptoms, okay. So, but this work is from Nature or Science, I'm not sure.
It's a recent one.
It's from 2019. So it's really recent, and more data is needed to be available to prove this link. Of course, we are not trying to inhibit completely
this
the
process,
enzyme. Okay?
Because as any other biological system, if it is there, maybe it is because it's necessary. It's like the oxidative stress. It has a biological role and function in our cells. But if it's highly increased, it is harm. Okay, it does harm to our cells. But if it's normal, it's normal. So,
Yes.
like nitric oxide, it has a physiological role. But of course, if it is very increased, it will be pathological and not physiological. Just in this case, the same thing. So the mechanism exists because it clears some toxic products. But of course, when it's too much, it's too much. Okay.
Yeah, and that's why it is so important. I'm going to pass my next slide. It is so important to not only to be able to say, okay, our extracts are able to inhibit in a dose-dependent concentration, but it also is important to deepen the knowledge on how our extract can perform that kind of inhibition. Okay, so we have also been conducting some kinetic experiments with the most promising extracts or compounds, okay? And as you can see, and this was not very surprising, of course, because we have an extract, which is a mixture of compounds, a purified one, okay? But we have several compounds acting maybe by synergisms, okay? So we have achieved a mixed type inhibition behavior, but this kind of information is really important, okay? It's really important to say that our extract is able to modulate by acting through this mechanism. So the compounds in our extract can bind either to the enzyme or to the complex that is formed between the enzyme and the substrate. Okay, it's really important to characterize this kind of behavior.
Okay, so of course, when we want to test our hypothesis, we need to test it in several ways, and whenever pertinent, in models as close as possible to a living organism. So in addition to these in vitro assays, we also use cell-based assays. Logically, animal models or clinical trials are the most reliable ones. However, unfortunately, there is a lot of bureaucracy that is needed to justify their use. So before you start the cell-based assays, there are some key points to consider. Is it necessary? Because they are very expensive, they are time-consuming, and you need a lot of know-how and expertise to interpret the results. And also, do you have the necessary materials and the facility conditions? Because everything needs to be sterilized. And then the cell models. How can I choose one? Okay. So in our laboratory, we have this model, this cell model. That is a DV2 mouse microglia cell line. And we use it to test the anti-neuroinflammatory activity of our samples. So in a very brief explanation, these cells, when are insulted with interferon gamma, they respond by increasing NEO production. And we already discussed the importance of this marker, okay? So in the other so if our cells are pre-incubated with the test samples, and then interferon is not able to increase the nitric oxide response, maybe it's a good signal that these samples will help to mitigate the neuroinflammatory response of these cells. Now you can. Okay, so when you are working with cells, you have also to ensure that the culture conditions are always maintained, okay? And it is mandatory to always check cell morphology before, during, and after the experiments. Me and Mariana, when we arrive to the laboratory, not now, but other times, the first thing that you do and the last thing that you do is check the cell morphology, okay? So, as you may understand, of course, before you test their anti-neuroinflammatory effect, we need to verify if our samples are not cytotoxic to these cells. One assay that you can perform is the Mtt Viability Essay. Okay, is one of others, and it can help you to test this. And of course, we will only proceed to evaluate the potential of our samples in concentrations that do not harm the cells, okay? At least in terms of MTT viability. And you can see in this work that we tested 2 extracts, but only one was able to reduce nitric oxide release. And we construct this type of graphs that you see here because that clearly helps you to interpret the results. However, at naked eye, we could incorrectly assume that at certain concentrations in the Tecatiga extract, we have a positive effect, and that is not true. And that is the reason why it's vital for you to perform statistical analysis of the data that you collect.
There's a point in your work, of course, where you will have to bring it all together, put it all together, okay, and try to create a storyline for your results. We believe this comic is our mantra, okay, is a real representation of our daily work. And in fact, is also the representation of this communication, because we are fully aware that we were supposed to talk only about abstracts, okay? But how can we do that in 1 hour and a half without showing you the work that we've been behind the single writing of an abstract, okay? So, as João was saying, gathering and statistics can help gathering and interpreting the results. In here, I show you some statistical tools like correlations, in which we were able to correlate the total amount of phenolics with the several bioactivities we explored. But for instance, in principal component analysis, we have a normalization of a larger number of variables, and we can establish different relationships between, for instance, the chemical composition, both in terms of qualitative and quantitative profiles, and the bioactivities. So in here, we were able to establish 3 groups. And the conclusions in here were like the compounds that were behind, or possibly they were behind the observed bioactivities, were those from these samples, which were the tetramers of fluoroglucinol. And this kind of information is really valuable to summarize all your work and put all that into an appealing abstract. And we have another question, and let's just check. Okay.
I think maybe another explanation.
I'm not yet, okay.
The sound is not okay?
No, the sound is perfect.
Okay.
maybe the explanation.
Maybe it's... Are you alone?
Okay, we'll try to do maybe a little slower,
but it's almost time.
Okay.
Yeah, we are almost reaching the end of the presentation. We apologize if our English was not that good, but I think. Okay, thank you, Frankie. See you later.
I think Alan has another question. Alan,
Really?
you can.
Let's check.
He raised his hand.
Hello, good evening, and sorry. Your English has been very understandable, and you don't have to worry about it. But what I want to ask about is we have been working with Excel to process data, but are there any other software that can process data more efficiently and give prettier graphics for our research? And are there...
Your sound is off, Alan. Okay, but I will.
The first part of the question we can answer.
Yes.
I'm sorry. Did you get my question? I think that
Only the
voice
first
was
part.
cut out.
Yes, only the first part.
Can you hear me?
Yes.
Hello?
Do you prefer to write on the chat?
Yes, of course.
Okay. But I can answer to your first question. We do not usually use Excel. We use Excel only at the initial state.
Because the instruments that we use are normally connected to Microsoft Word and Excel and those stuff. So we initially, to get the raw data, the absorbance reading, for instance, yes, we used Microsoft, we used the Excel. And it's a good way for you to start a table and to make a graph, to plot a graph, it's okay. But then when we want to make things more beautiful, and keep in mind that researchers are not illustrators or web developers or nothing like that, but we try to do our best in that point because we like to make our presentations appealing. Yes, we use the GraphPad, for instance. This is not publicity because we don't sell.
No, you have to pay for
and you have to pay the license. But yes, we use Graph Abstract. And it's very good because, GraphPad, it is a very good software because it makes it possible for you to put all your data and, for instance, plot graphs and also make statistical analysis. So that is one.
SPSS.
SPSS.
Yes.
Sorry, could you please write it on the conversation because I didn't get it.
I'm trying to.
Just a moment.
Okay. I cannot.
Maybe if you first put an I so we can have the chat box.
It's in here.
Okay, but
no, it's in here.
Okay,
it's in here. Okay. So it's GraphPad and SPSS.
There may be another ones, of course. These are the ones that we use in our lab every day.
Mafalda, are you aware of a software? It's called, I think, Statistics.
Also see.
Right? Because it's available for students in
the Port University of Port. Also, you may check.
Yeah, the second one, I know it. Yeah.
Okay, and statistics also. I'm just trying to check if the name is correct of the software. Because we only use the GraphPad one and SPSS.
I don't know. I don't.
Okay.
The thing is, you can always check with your team or the informatic if they have some licenses for other. Okay, here in our university we have some. Actually, GraphPad Prism we also have to pay for it, but we have some free versions of some software. Okay. So returning.
Sorry.
Yeah, we lost connection, Alan. But maybe you can put on the chat, it's easier, maybe. Okay. So, can I proceed? Yes? Okay, so at the end, of course, you should be able to draw conclusions from your work, okay? And of course, be aware, conclusion is not the same as an abstract, okay? Besides both being short texts, okay? The most remarkable difference between an abstract and a conclusion is that an abstract, as I was saying, is really factual and concise, and in the conclusions you can have your perspective, your future perspective or future conclusions. You have here the example of that is the same article, okay? I forgot to put the reference of my article, but it's the same article, and as you can see, the abstract section and the conclusion section is completely different, I think. Because this one summarizes your results and also opens door for future research, and also highlight some weaknesses or some strengths of our work. The abstract, as you may know, is gives only the main findings, the key findings of your research work.
To catch the attention.
Yes.
So, okay, the final step of every work in academia is that after writing your report, you have to subject it to peer review. Of course, in your case, it can be your professor or someone with more expert in the field, someone expert in the field. Or even a colleague. Me and Mariana often shared our papers for each one to try to see some mistakes or some opinions that could be helpful in each other's works, and it worked very good.
Yes.
Okay, sometimes it's hard that after all the hard work and dedication, you will receive some critiques, bad critiques or good ones, or also some rejection of your ideas and results. But don't despair, okay? This is a normal process for everyone in science. And very often you can learn a lot with the opinions of others, okay? So we have here some examples. These are some, we have some questions.
Yeah.
Yes, so the first is from Alan, saying, can you please recommend introductory books or online courses for students interested in pursuing research?
Okay.
We will have to check it because...
in fact, I think it was our personal convictions that
we shared.
Yes, because I've always wanted to be part of research, of the scientific community.
And we started very soon,
yes.
And we were lucky to start in this lab when we were students, pharmaceutical students. And some of the knowledge was passed to us by the elder members of the group. So we never actually
needed to
check some books because we had those persons to guide us and to help us. Of course, you have that's another question that to share the email addresses, we can put it here. We also have it in the first slide of the presentation. But if you want to ask some of these things, yes, we can check if you want some type of books or some type of research lines to help you.
Yes, but be aware, this is not, we are not the rule, okay? We are only giving you some advice.
Yeah, some advices and
we were
our experience.
Students, yes, we were students. I joined the lab in my 4th year. João joined in his 3rd year. Yeah, so maybe you can, if you are in a university, of course,
instead of reading a book, maybe you
can talk with the professors, show your interest,
and ask for the opportunity to collaborate with them. We also receive students from very early stages in the formation and they come some hours or days with the laboratory with us, and they follow us in our tasks, how to prepare solutions, reagents, how to test compounds, and this is the type of exercise that we do with them. So maybe you have some groups in your university that are also able to help you with that. Of course, there may be some information already published about this, but if you really have the opportunity to engage in a team, in a research team, maybe it's more profitable to you because each group has some form of doing research and to ask for new answers to their problems. So maybe it's better for you to try, okay, that way. We are just writing our emails for you. Yes.
No. Okay, so yes. I don't know what's happening.
So you can contact us if you want.
No, the emails are not correct. I don't know what's happening with the chat. Sorry, don't consider these emails. I don't know what happened. Okay. Yeah, this one here is mine, and then João Bernardo's. Okay, now the last two are correct. So sorry, but yeah, I think it's really. I think talking is always a good way to learn
and experience
yes.
with the research team. Okay, so just to finish, we were talking about peer review, and we have some comments that were from random reviewers after we submitted our own works for publication. And sometimes you see that reviewers can make very helpful suggestions. In this example, they suggested to introduce new keywords. And this is very relevant, okay, just as the abstract keywords are relevant. And why? Because they represent the search queries that people use in search engines, okay, as the ones we talked about, like Google Scholar or Scopus. And and in other example, they also acknowledge your methodologies, but suggest that next time you should use more suitable ones. And it is okay for them to give an opinion. And maybe it's another way for us to also learn about other techniques that we did not know. And now with this information, we can look for it and try to implement these new techniques in the laboratory.
Of course, we expect that reviewers are chosen by their expertise in a particular subject, in a particular field of knowledge. So when they point some weaknesses or strengths of our work, they also point relevant flaws. And sometimes we have to acknowledge and recognize that our work is an ongoing work, okay? It's not only always a final work. Sometimes you have to do more experimental work to better support your conclusions. You have here the example of a review that was made to a paper, to a work by me, and I had to perform 24 more analysis. So I could better support and improve my chances of publication. So this is not a bad thing, and most of the times they really want us to explain better our experimental findings, our choices, and they stimulated us to compare our results, to do better. Okay. So we have to be able to deal with this kind of criticism and facing it as a continuous opportunity to learn, of course. And that is exactly what we intended today with this workshop, to share with you what we have been learning in our everyday. We still have been learning.
Always learning.
Always learning. So we hope you enjoyed as much as we do. This time we spent together. We have to thank, of course, the International Pharmaceutical Students Federation for this invite, and extend, of course, our acknowledgements to our scientific coordinator, Professor Paulo Andrade, which gave us our support, of course. And thank you all. And now, Mafalda, I give you the spotlight.
I would like to thank you again for your presentation. I think I speak for everyone when I say it was very clear and relevant. I think Alan wants to make another question. Alan?
It's not really a question, just saying thank you very much. It was very interesting for me to discuss this topic with you. And I'm very looking forward to new webinars because it was really interesting and it answered a lot of my questions. Thank you very much, doctors.
Thank you.
We also have Gaston. I don't know if I'm saying your name correctly, but you can have the word.
Did you? Oh, sorry. It's not my part. I'm sorry.
Maybe you can write it in the chat. Alan, do you want to ask another question? No? Okay, so I'm passing the word to Emin from the... Yeah, you can have it.
Okay, thank you, Mafalda, and hello everyone. My name is Emine Merve Yıldırım, and I'm the Pharmaceutical Sciences Initiative subcommittee member at IPSF. So first, I would like to thank our speakers for their presentation, and it was an amazing, informing workshop. And I would also like to thank IPSF EuRO team, and especially Mafalda for this great workshop and for collaborating with us under this initiative. And as you know, as IPSF team, we prepared an interregional research project for you to learn more on research, which includes 3 webinars and 5 workshops. And this workshop was the second workshop under this initiative. So if you enjoyed this workshop, you can join in our second webinar on project design, drafting ideas, and collecting data, which will be held on this Friday, 1 p.m. GMT+1. You can find the registration form in the IPSF social media accounts, and stay tuned for our following events. And I also would like to thank you for your participation. Thank you for joining me in this workshop. And thanks again for our speakers and for IPSF EuRO team. Okay, you can have the floor, Mafalda.
If anyone has questions, if not, I think we can end the workshop. And thank you once again for coming.
Thank you very much.
Thank you.
Thank you so much.
Bye.
If you want to say goodbye in your native language.
Me.
كده ciao.
يعطيكم الصحة وميرسي بوكو. شكرا كثير. كاين بزاف لغات في البلد تاعي. There are many languages. Thank you very much.
Thank you.
Obrigada.
So I'm ending this session for everyone. So thank you again for your collaboration, and bye.
Bye. Adeus.